Welcome to This Explainer. Today, we're unpacking the absolutely fascinating, strictly mechanical story of how life copies its ultimate blueprint. Think about it. Right now, literally every single second, millions of your cells are actively dividing. But before any of them can actually do that, they have to copy their massive genetic instruction manual, and they have to do it perfectly. So we're going to look closely at exactly how that microscopic machinery operates. Here's our roadmap for today. First, the blueprint of life. Then, the rules of replication. And finally, 3 historical models. Kicking things off with Section 1, the blueprint of life. So before any single cell can divide into 2, it's absolutely crucial that it flawlessly duplicates its entire genome. We call this process DNA replication. It all starts when the original double helix literally unzips itself. Once it's unzipped, those single strands step right up to act as perfect templates for constructing completely new, complementary strands. It's just an incredibly precise method of duplication, making sure the new cell gets the exact same genetic manual as the original. Moving right along to Section 2, the rules of replication. Now, biology doesn't just guess here. There are strict laws of chemistry at play guaranteeing an identical copy every single time. The nitrogenous bases of your DNA only fit together in highly specific ways, and this is all governed by hydrogen bonds. For instance, adenine always pairs with thymine, locking together with exactly 2 hydrogen bonds. On the flip side, cytosine pairs with guanine, forming a slightly stronger molecular handshake with 3 hydrogen bonds. This strict matching system means if you have one side of the unzipped DNA, you automatically know the exact sequence of the missing half. It's like a giant puzzle where only specific pieces fit together. But the actual construction, well, it isn't a free-for-all. The enzyme responsible for building the new DNA, which is called DNA polymerase, acts kind of like a highly picky factory worker with 2 unbreakable rules. First up is primer dependency. It absolutely refuses to start from scratch. No way. It requires an RNA primer synthesized by another enzyme called primase just to kick off the whole shebang. The second rule is directionality. This picky worker only ever moves and synthesizes the new strand in one highly specific direction, 5' to 3'. It literally cannot work in reverse. Because the 2 original unzipped strands of the double helix run in opposite, anti-parallel directions, our picky factory worker really has to adapt. On one side, it's a total breeze. It creates a smooth, continuously synthesized leading strand, mainly because it's moving in that natural 5' to 3' direction as the DNA unzips. But on the other side, well, it has to work backwards, away from the unzipping fork. This means the lagging strand can only be synthesized in short, fragmented bursts. We call these little fragmented bursts Okazaki fragments. Honestly, it's a beautifully complex workaround to a very strict biological rule. You know, it's super easy to take this elegant, complex machinery for granted now that we've mapped it all out. But let's pause for a second and ask, how did early scientists hypothesize this microscopic copying actually worked before we understood these strict rules? I mean, just imagine trying to figure this out before the era of modern molecular biology. Which brings us to Section 3, 3 historical models. Historically, the scientific community fiercely debated 3 distinct models to explain how the DNA double helix manages to replicate itself. They argued between semi-conservative replication, conservative replication, and dispersive replication, and each of these offered a radically different vision of what exactly happens to the original DNA molecule during that duplication process. What's really fascinating to think about is the stark contrast between the 2 incorrect theories. Let's consider conservative replication first. This model assumed the original double helix remained entirely intact. It essentially acted as a closed mold, or template, to churn out a totally separate double helix molecule made of 2 brand new strands. Then, on the complete opposite end of the spectrum, was dispersive replication. This theory predicted a rather chaotic process where the parental double helix was basically chopped up into fragments. Each fragment served as a template, and the end result was a messy mosaic of old and new segments scattered completely across both strands of the resulting DNA molecules. Ultimately, though, brilliant experiments proved those extreme ideas wrong and confirmed the elegant, semi-conservative mechanism. And this is the confirmed standard for literally all known living organisms on Earth. When those parental DNA strands separate, each one serves as a dedicated template. Therefore, every newly synthesized double helix is perfectly composed of one original parental strand and one newly constructed daughter strand. It's the perfect middle ground. It's efficient, exact, and beautifully simple compared to the alternatives, half saved, half entirely new. As we wrap up this explainer, I want to leave you pondering a truly mind-bending thought. If this exact semi-conservative unzipping and copying, navigating all that strict base pairing, the leading and lagging strands, and those picky RNA primers, if all of that is happening flawlessly inside your body billions of times right at this very second, what other biological marvels are hiding in your cells just waiting to be uncovered? I mean, the deeper we look into the mechanics of life, the more extraordinary it becomes. Thanks so much for joining me, and I really hope this sparks your curiosity to keep exploring.